Welcome everybody. Now we will continue our lecture. We have started a series of lecture on nervous system. So last time we have studied about the synapses. So today the topic is very important because today we are going to deal with the development of neural system. How the nervous system develop from embryonic stage. So today we will focus on the development of neural system. So let us start. So we will go with the very basic concept. Suppose I will start from the fertilization. So in the fertilization, this is your ovum and this is your sperm. This ovum and sperm when fused together, then it is the formation of zygote. So this is female pronucleus and this is male pronucleus. I think everybody know the concept of fertilization. So little bit review here, because from the starting, if you deal from here, then it will be easy to understand about the development of neural system, because it is not as much easy that you can see here. But when we'll go by the embryonic stage, from the initial stage, then it will be easy to understand that how the development of neural system take place. So this is the fusion of sperm and ovum, and this is the zygote having diploid number, and here the fertilization takes place, that is called zygote. This is the zygote. Now this zygote, this pronucleus and male pronucleus, they fuse together, and when they fuse together, this is now diploid. 23 chromosome from female and 23 chromosome from father, from maternal and from paternal. This is when we study about the embryology, then I will go in the big detail. So this is the zygote, and this zygote, this divide into two cell stage, and this stage from 2 to 4 cell stage, and this stage is called blastula. What is called? Blastula. And after 4th stage, again these cells divide and it will go to the 12th cell stage, and that stage is called morula. It means that if you see that when cell divide, it undergo a smaller entities. These cells are smaller entities, and after morula, then again this cell divide, and then this cell rearrange inside the cell, and this rearrangement in the central, the cells are called inner cell mass, and the outer are called outer cell mass. So fusion and zygote, blastula stage and morula stage, and after that there is a rearrangement of cell is going on. And then again this step is very important, that this inner cell mass will go to the periphery, to the anterior side, and this anterior side, and this is the outer cell mass. So from here, now from the inner cell mass, your neural system will be the formation of neural system will take place. So this point is very important. So here what happens, that now these cells rearrange. These cells rearrange, and it will form two layers here. One layer, this is called ectoderm, and this is called endoderm. It is bilaminar disc. Now this cell, this will form the bilaminar disc. From inner cell mass, this cells, because I am not going to the detail, that is amniotic cavity and the yolk sac. Only I want to say that now this cells is going to rearrangement, and this will undergo the formation of bilaminar disc. You know, bilaminar disc is just like one disc is here and second disc is here, like this. Like this is called bilaminar disc. This is called bilaminar disc. After that, these two discs here, these two discs tightly held with each other. This anterior side, this anterior side, this anterior side, you can see here, this anterior side is very tightly held. Both the discs are very tightly held together. They are joined together very tightly. You know, this area where these two discs are pressed and tightly tied, this is called precaudal plate. This is called precaudal plate. It means that from here the real mechanism or real formation of neural system will start. This is the point from where, because now I am going to make our neural system from here. So these two discs, bilaminar disc, when we will reach up to bilaminar disc, this disc is actually, this is here tightly they are held together, joined together, and this is called precaudal plate. This is called precaudal plate. This is called precaudal plate. Now move forward. Now this point, actually what happens here, the cells are degenerated. You know, here few cells are degenerated longitudinally, and when these cells are degenerated, they are undergo degeneration. When these cells are degeneration, so there will be the gap. See, these cells are undergoing degeneration. These cells are undergoing degeneration. These cells are undergoing degeneration longitudinally toward the procaudal plate, and this is actually this degeneration. Now this area is called primitive node and this is called primitive streak. This is called primitive streak. Very important point here, very important. Now this precaudal plate later on it will develop into the mouth. This is the anterior portion. It means that this area is your cephalic area. Cephalic means head area, head and neck, and this is your caudal. Caudal means tail. Caudal area. Caudal area here from the cephalic end, it means it is the mouth, and this is the caudal area where the cells undergo degeneration process, and see, it is a longitudinal streak, and that is called primitive streak, and this is the primitive node. This is the primitive node and this is the primitive disc. So in the bilaminar disc, the formation of primitive node and primitive streak has been started. Now another point, very important point. When these cells, see, when these cells, here you can say, just for to understand, see, suppose these cells undergo degeneration, now from here cells move internally from here. It will go inside, and this cell will go inside. When this cell will go inside, under this ectoderm and endoderm, the middle layer formation take place, and that middle layer is called mesoderm, and this is the formation of trilaminar disc. This is the germinal layer. So this is the trilaminar disc. Ectoderm, endoderm, and in between the ecto and endoderm, this is the mesoderm. It means that from this primitive streak, the cells undergo inside. These cells undergo and take the formation of mesoderm. It is not very complicated. It is very easy. Just try to understand. It is the only rearrangement of the cell, where the cell is going, how the formation of your neural system take place, how the development of baby occur. So this will go undergo, and there is a formation of mesoderm, and now this is your primitive streak, and this is a primitive node, and this is your procardial plate. It is just similar what exactly we have explained here, what I have explained here. Okay, this is a trilaminar disc and this is a bilaminar disc. I think from up to here everybody knows. Now this area, you cut this longitudinal section here, cut. I am going to explain now this area, how the neural tube formation take place. So here it is very easy. This is your procaudal plate, this is your procaudal plate, this is your primitive node, and this is your primitive streak. Here what happens, again the cells move forward here, again the cells move forward, and these cells move forward further, this move, and these cells move forward, and this will go like this, this will go like this, see, from notochord, and this notochord, this is from primitive node, and here from the primitive streak, this cells proliferate and move forward to the cephalic end, and here it will cover. This is the anterior pore and this is the posterior pore, and this is called your neural plate. This is called neural plate, neural plate. And here this is called your notochord. This is called notochord. I think it is not difficult. Formation of trilaminar disc. This is your notochord, this is your primitive node, this is your primitive streak, and this is your procardial plate. This is structure, flip in the anti-clock. This is structure, and this primitive streak, primitive node. From here you understand how the degeneration occur. When degeneration occur, primitive node and primitive streak formation take place. Now the cells move inside, mesoderm formation take place. Now from here cells also move toward the cephalic end, toward the procardial plate, and when this move to the procardial plate, see here, when it moves to the procardial plate, there is a formation of notochord, and this around notochord, actually then what happens, this notochord, this is undergo depress, this depress, and this edges will raised up, this edges will raised up. It is just like that, this is just like that. Suppose from middle here, this is your middle here. Suppose if I can talk like this, suppose this is your middle layer. When this middle layer is depressed, go down, then it will come upward. So this is actually, when this come up, this is a neural plate and this is a notochord. Now this area is called notochord. After that, another important point here, this cells, some cells are detached from neural tube. These cells are detached. When these cells are detached, the cells will detach from the notochord. The cells which is detached from the notochord, these cells are called neural crest cell. So here what we can say, this is your endoderm, this is your endoderm, this is your mesoderm, okay, and this is your notochord, See, this notochord will in future it will make brain and spinal cord, and these are the neural crest cell which are detached. Some cells are detached. It will go lateral side from the notochord, and this is called neural crest cell, which will form other parts of the brain and spinal cord. Neural crest cell. This is mesoderm, and this is ectoderm. This is ectoderm, and this is endoderm. So basically from the ectoderm, you can see this is the layer ectoderm. That's why it is called neuroectoderm. Why it is neuroectoderm? Because the origin of neural system is coming from the ectoderm. That is called neuroectoderm. That is called, the term is called neuroectoderm. Neuroectoderm. Ectoderm. So it is very brief explanation about development of neural system. See, from two cell state, blastula, morula, inner cell mass and outer cell mass, then rearrangement takes place. Bilaminar disc, this will tightly held. This is procordial plate, which is the anterior part. This is the anterior part which will undergo mouth in future, and this is your primitive node and primitive streak. And then it will undergo, the cells will move inside. When the cells will move inside, then the formation of mesoderm take place. Now the trilaminar disc, and after that these cells will move forward like this. These cells will move forward, and when these cells will move forward, it will take the formation of notochord and take the formation of neural tube. The formation of neural tube, notochord, it will joint like this, and this is your notochord process, and these are the neural crest cells. Some cells are detached, fall adjacent to the notochord, and these cells are called neural crest cells. So this is your diagrammatic representation. Representation. So by this we can say development of neural system, it take place by the primitive node and by the primitive streak, from the primitive streak. Now next point, primitive streak. Okay, so like in a simple way, in a very simple way, what we can say, in a very simple way we can say that, in a very simple, so this is your it means that now this is your neural plate. This is your neural plate. This is your neural plate, and this is your mouth. This is your mouth. This is very simple what we have studied. So if you can say this is the neural plate, and this is our neural tube, and this is the mouth. From here, the formation of vesicle take place. It means that this will form the vesicle, and the vesicle is like this. The vesicle is like this. Three primary vesicle, formation of three primary vesicle take place. Formation of three primary vesicle take place. Formation of three primary vesicle take place with a different color. Let us put this color. So from this neural tube, three primary, this is called three primary vesicle take place. Three primary vesicle. This is called primary vesicle. This is called three primary vesicle. This is called three primary vesicles. Three primary vesicles. This is called three primary vesicles. Okay. Now they have different name. This vesicle, the first vesicle, this first vesicle is called prosencephalon. This is called prosencephalon. Prosencephalon. Now the second is called mesencephalon. Mesencephalon. And the third vesicle is called rhombencephalon. This is called rhombencephalon. This is called rhombencephalon. So another name is given, prosencephalon means this is also called forebrain. This is your forebrain. Forebrain. This is your forebrain. Mesencephalon, midbrain. Rhombencephalon is called hindbrain. Hindbrain. See how the neural plate formation of three vesicles take place, and you will see later on how the central nervous system development take place. Now other point which is very important, you need to remember that from this forebrain, later on this forebrain, in the forebrain there will be olfactory lobe, area, different area, olfactory lobe, cerebrum, and the structure which is present on the forebrain, important structure, and then diencephalon. This you need to remember. Later on I will go in detail. And midbrain, optic lobe and crura cerebri. Optic lobe and crura cerebri. So this two part is present on midbrain. Now the rhombencephalon, the important point here, the rhombencephalon, if you talk about the rhombencephalon, very important point, pons cerebellum and medulla oblongata. Okay, so now formation of three vesicle. First vesicle is probencephalon, forebrain. The structure are olfactory lobe, cerebrum, diencephalon. This is the part of mesencephalon, that is called midbrain, optic lobe, crura cerebri, and this is the rhombencephalon, that is hindbrain, and this is structure heavy in the hindbrain. You will see pons, cerebellum, medulla oblongata. Now after three primary vesicle, there will be a formation of after three vesicle, there is a formation of 5 primary vesicle. There is a formation of 5 secondary vesicle. This is a primary vesicle and this is the 5 secondary vesicle. So here actually what happens in this area, here this portion will be remain constant. It will not enlarge and it will fix. It will not go. But this portion will be further, it will spread. This portion will be further enlarged. this portion. This portion will be a further move forward, this portion. It means that if in a big picture you can see here that how to form the, this is okay, and now from this is a fixed portion. Now after that it will divide into two. It will divide into two. It will divide into two. Let me put in a proper perspective. It will divide into two. It will divide into like this, and this is your smaller one. So this forebrain, it again divided into two, and after that, so let me put in a same color. This is red color, so this will, and this is this. And then after that, 3, 2, 3, 1, 2, 3, then again this will be one more color. And then your, so this is your 5 vesicle. This is your 5 vesicle. This is your 5 vesicle. 1 vesicle this, 2, 3, 4, 5 vesicle. This is a secondary 5 vesicle. Actually what happens, now let me name it. Now this is actually this is called, so this is called telencephalon. 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 Telencephalon. And this is called diencephalon. Diencephalon. This is called diencephalon. Then after that, this is diencephalon. Then your midbrain. Now this is your midbrain. This is your midbrain. This is your midbrain. Clear? And now after that, this is called metencephalon. This is called metencephalon. Metencephalon, and this is called myelinocephalon. So from the first vesicle, see, from the first vesicle, 3 primary vesicle, this is divided into 2, telencephalon and diencephalon. This is very smaller, and after that it will enlarge, and this will form telencephalon. And this is the midbrain. Just like here, mesencephalon is the midbrain. The important point here, the midbrain, the back of the midbrain, there is one more formation of vesicle take place on the back side. These cells proliferate, and this is called cerebellum. This is called your cerebellum. This is called your cerebellum. Very important structure, cerebellum. And cerebrum is here. This is the cerebrum, and this is the bellum, cerebellum. This is called cerebellum, and after that 2 more structure from the rhomencephalon, that is metencephalon and myelinocephalon. From here, metencephalon and myelinocephalon, from where pons, cerebellum or medulla will take place. So I think it is very clear. This is neural tube. We have seen how the neural tube formation take place. This is the mouth, and now this will be the anterior portion, the posterior portion. Now from here, this portion, anterior portion, which pore is joined together, and this is the posterior pore, they join together, and anterior pore join at the 25th week, and posterior is joined at 27th week. But anyway, now three primary vesicle, then three primary vesicle, proencephalon or mesencephalon and rhombencephalon, and from this primary vesicle there is a formation of telencephalon, diencephalon, this is the midbrain, metencephalon and melanocephalon. So this is the 5 secondary vesicle formation take place. So now we are moving to the development of neural system, that is brain and spinal cord. Actually means what is brain? Actually brain is to command and control all the system. It is the central organ processing system of our body. It controls everything. That is command and control system. And second point, actually 70 to 80% the brain is developed up to 2 years, but completely the brain is developed up to 6 years, and the spinal cord fully developed up to 4 to 5 years. 4 to 5 years. Now after this, we will, after 5 secondary vesicle, after 5 secondary vesicle, let me draw. So this is actually, this section is like this. Now I am going to draw anterior and posterior. Suppose I am going to draw anterior and posterior. Okay, anterior and posterior. So anterior and posterior. Now this is the midbrain. This is the midbrain. This is the midbrain in a different color. Let me draw in a different color. So first of all, this structure you need to remember. This structure you need to remember. That is your, this is your structure. This is your structure. This is your structure. This is your structure. Then after that, okay, and then again okay, and so midbrain like this, and this will move together, this will move together. Okay, and here the important point is that from back side, this is your cerebellum. This is your cerebellum. Okay, so importance, this is your midbrain, okay, this is your pons, and this is your medulla oblongata, medulla oblongata. Okay, this is your medulla oblongata, and you know, after that, this is your diencephalon. This is your diencephalon structure. Suppose this is your structure, diencephalon. This is your structure, that is diencephalon. This diencephalon. Now the telencephalon. So what happens that this telencephalon, now it will move forward. Some structure it will be underdeveloped, but what happens, this will go and this will go, and this will go, and you can see this is your midbrain, this is your diencephalon, this is your diencephalon, and this is your, now this is your forebrain, this is your forebrain of frontal lobe, this is your frontal lobe. This is your frontal lobe. Frontal lobe. Frontal lobe. This is your parietal lobe. This is your parietal lobe. Parietal lobe. Parietal lobe. This is your occipital lobe. This is your occipital lobe. Occipital lobe. This is occipital lobe, and this is your temporal lobe. This is your temporal lobe. This is your temporal lobe from 5 secondary vesicle. So see how this brain will develop. And if you want to make it more clear, this is your mouth, and this is your frontal lobe, parietal lobe, occipital and temporal lobe. So now you can see here, first of all, morula, zygote, morula, blastula, then 2 laminar discs, then mouth, and after that primitive node, primitive streak, and then after that notochord, neural plate, and that neural plate is converted into 3 primary vesicle: prosencephalon, forebrain, mesencephalon, midbrain, romencephalon. 3 primary vesicles converted into 5 secondary vesicles: telencephalon, diencephalon, midbrain, metencephalon, and melencephalon. And this is your, in between this, you can see in between this is your, this is your cerebral spinal fluid. This is your cerebral spinal fluid. This is your cerebral spinal fluid. But anyway, now from this telencephalon, this will diencephalon, and this will enlarge more, and this will converted into frontal lobe, parietal lobe, occipital lobe, temporal lobe, diencephalon, midbrain, pons, medulla, and this is your cerebellum. This is your cerebellum. This is your cerebellum, and this is your, all this part are called cerebrum. Cerebrum. So see, I am standing here. This is your nose. This is your, and this is your brain. One lobe is here, and second lobe is here, connected with the corpus callosum. We will go into the detail. But here, just I want to tell that here, by this way, the formation of development of neural system take place. So now I am going to stop lecture here, and next time we will go in the different structure of the brain, and I request you to please subscribe, and thank you very much.